Hello students, I am S. Gejalakshmi, working as PG Assistant Microbiology in Government Girls Higher Secondary School, Redhills, Tiruvallur District, welcoming all to this video lesson. In last video, we understood what is Hybridoma technology. In this video, we are going to see how HATmedium is used for selecting Hybridoma cells. If you missed Hybridoma technology video lesson, no worries. I have given its explanation in both Tamil as well as English link in this particular video description part. You can see it. Okay, let us now get into the video lesson to know how Hybridoma cells are selected in HATmedium. In order to understand the principle behind HAT selection of Hybridoma cell, it is important to know that whenever mammalian cells divide in artificial media, before dividing, it should synthesize a new copy of DNA. For that, it requires a synthesis of nucleotide. What are nucleotides? They are the basic structural unit of nucleic acids such as DNA and RNA. Mammalian cells synthesize these nucleotides using two different pathways. They are de novo pathway and salvage pathway. Under normal condition, mammalian cells will use de novo pathway to replicate. When de novo pathway is blocked, cells uses salvage pathway as alternate. But this only happens when hypoxanthine-aminopterin-thymidine are present. This key approach is used in HATmedium to select Hybridoma cell. In HATmedium, cells cannot operate de novo pathway, and this is due to the presence of aminopterin in the media. This aminopterin blocks one key enzyme that is required to operate de novo pathway. The enzyme that is blocked by aminopterin is dihydrofolate reductase. When this enzyme is blocked, cells do not able to operate de novo pathway. Therefore, the cells have one option to work out, that is salvage pathway. In order to operate salvage pathway, cells should contain the enzyme called HGPRT, that is hypoxanthine-guanine-phosphoribosyltransferase transferase. The cells which has HGPRT enzyme can utilize hypoxanthine-aminopterin-thymidine in the HATmedium for the synthesis of nucleotides. When we look after the condition of plasma cells and myeloma cells have this enzyme but myeloma cells are not. Yes, the myeloma cells used in Hybridoma technology are mutated. Two genes are mutated. One of the gene is mutated in HGPRT, that is, they cannot produce this enzyme, so the cells cannot operate salvage pathway for synthesis of nucleotide. Another gene which is mutated is they cannot produce antibodies. So the myeloma cell used in Hybridoma technology are HGPRT minus and IG minus. When antibody producing HGPRT plus plasma cells are fused with immortal myeloma cells, Hybridoma cells are produced. Along with the Hybridoma cells, fused and unfused plasma cells, fused and unfused myeloma cells are also being produced. Among these cells, Hybridoma cells alone has to be selected. So now the HATmedia as aminopterin. So the cells cannot utilize de novo pathway for the synthesis of nucleotides. So the cells has one option to choose, that is the salvage pathway. In this condition, fused and unfused myeloma cells cannot able to live or survive in HATmedium because the myeloma cells are HGPRT minus. In case of fused and unfused plasma cells, these cells can survive in HATmedium because they have this particular enzyme called as HGPRT, so they can utilize salvage pathway and synthesize nucleotides. But plasma cells have short lifespan, so automatically they will die within a short period of time. So the only leftover cell in HATmedium is Hybridoma cell. Hybridoma cell has both the property of plasma cell as well as myeloma cell. That is, the plasma cell has HGPRT enzyme, and myeloma cells are immortal in nature. So these hybridoma cells are selected for the continuous production of monoclonal antibody in the HATmedium, and this is the principle of HAT selection. Hope you understand this video. If you have any question, you just ask me. If you didn't subscribe the channel, please subscribe it and share it. Thank you for watching.